Preparation model paper 25. I'm Anand Shankar Reddy, PhD plant pathology. Kaira disease was first reported in. Actually, this Kaira disease was actually physiological disorder where we might have studied in rice, right? So rice Kaira disease was caused by. Rice Kaira disease is actually caused by zinc deficiency. They're asking it was first reported in. Terai district of Uttar Pradesh, Tamil Nadu, Bihar, and Madhya Pradesh. 4 options have been given. So this rice Kaira disease was first reported by I think Y.L. Nina and Padmanabham, probably 1970s, I think so. That was first reported in Uttar Pradesh, Terai district of Uttar Pradesh, where this rice Kaira disease was first reported. Black tip of mango was first reported. So Uttar Pradesh, Andhra Pradesh, Bihar, and Tamil Nadu. So this black tip of mango is actually uh, maybe boron deficiency and sulfur dioxide concentration is also two reasons are there. One is the boron deficiency and the second one is the sulfur dioxide concentration is also leads to this black tip of mango. So this was first reported from Bihar. So from Bihar where this black tip of mango was first reported. I think RS Vasudeva, I'm not sure, probably RS Vasudeva maybe is the person who actually reported it. I'm not sure, I repeat, I'm not sure. But it was reported from Bihar only. The only next question is the only plant pathogenic bacteria which thrives in anaerobic condition, not aerobic, anaerobic conditions they are asking. Ralstonia, Irvinia, Xanthomonas and Pseudomonas. Among the given options, this Irvinia can survive anaerobic conditions. The population of a host in which all the individuals have a particular character of resistance is commonly known as pathodim. So pathodim is generally refers to in which all pathogens have individual characters or particular characters in the resistance are commonly called as pathodim. So covering tomato nursery with a nylon net is followed for the control of. So in tomato nurseries covering this tomato plants is generally recommended for the disease called tomato leaf curl virus. So generally all plant viruses are mediated or transmitted through insect vectors, right? So this leaf curl virus is also transmitted through insect vectors. So to control the insect vectors is automatically leads to the controlling of virus. So just covering up this nursery helps to control this tomato leaf curl virus among the given options. Fungus producing a conidia either in Pycnidia or Acerulus belonging to which of the following? Hypomycetes, Coelomycetes, Blastomycetes, none of the above. So generally this fungus produces conidia either in Pycnidia or Acerula that belong to the this Coelomycetes. Principle of Fungal Taxonomy was written by, it is probably the book Principles of Fungal Taxonomy was written by Talbot, D.P. Tripathi, Ainsworth and Whittaker. Five kingdom classification was given by Whittaker, whereas Ainsworth gave his fungal classification probably in 1973. This is also one of the most followed recommending classification and following classification at nowadays. Ainsworth classification, Cricketal classification. So coming to the question, this principles of fungal taxonomy was actually written by Talbot. L.C. Coleman was a general plant pathologist during 1914, but worked and suggested the control measures for which of the following disease? Turwilt, blister blight of cocoa, Coliroga of areca nut, butterrot of pine. So actually retired L.C. Coleman, he was working as a plant pathologist during British time. In Karnataka, he just worked on a disease called Coliroga disease of areca nut. So the answer is answer C, Coliroga disease of areca nut. Extensive studies on black tip of mango disease was carried out by which of the following? Sadasivan, M.K. Patel, Das Gupta, and Bede. I think S and Das Gupta. Yes, it's not Vasudeva. Previously, I told Vasudeva black tip of mango, right? No, it's I think S and Das Gupta, probably 1958. I'm not sure. Probably 1958, where this S and Das Gupta was the first person who reported his black tip of mango is boron deficiency, and also this bricklings generally produces some kind of sulfur dioxide, right? So that is also one of the reasons for this black tip of mango. And S and Das Gupta was the person who actually worked on this black tip of mangoes. Citrus exocartosis viroid belonging to the genus. In viroids, only two genuses are there, Pospiviroid and Avicennaviroid. Generally, the Citrus exocartosis are belonging to the genus Pospiviroid. A genus of races, a group of races and biotypes of a pathogen species that can infect only plants within the certain host genus or species is called I repeat once again, a group of races and biotypes of a pathogen that can infect only within the certain host genus or species. It can only infect a particular host genus. For example, you can take Puccinia graminis tritis. Tritis only infect wheat, or pharma species like Puccinia graminis pharma species tritis that can only infect the particular host. That kind of things is what we can call as, it is called as biowar. Okay. The process of self-fertilization of a fungi is known as spermatization, automixis, somatogamy, and diphomixis. This is generally called as the process of self-fertilization is generally called as automixis. A classical example in controlling the plant disease by the use of suppressive soil is lead beta potato, red bramble, tur disease of wheat, tundu disease of wheat, and tecal disease of wheat. Generally, this tecal disease of wheat, we can control the disease efficiently by just you know, uh, by use of this suppressive soil. Generally suppressive soil in the sense by adding some kind of biocontrol agents, especially this wheat tickle disease that is caused by Gommonomyces graminis tritis. We can efficiently control by using a biocontrol called Pseudomonas fluorescens. S.D. Garrett was the person who worked almost for 20-30 years on this wheat tickle disease, and there is only one classical disease. There is only one example where we can control the disease 100% by using this biocontrol agent. That was also given by S.D. Garrett. We can generally consider S.D. Garrett as father of biocontrol agents regarding plant pathology. So this is a wheat tickle disease. Identify the wrong pair of virus vectors among the given options. Rhigas grass mosaic virus, if Eriophyid means Jejuni yellow mosaic virus, white flies, barley yellow mosaic polymyxa graminis, tomato spotted wilt virus, thrips. So among the given option, this probably this Jejuni yellow mosaic virus is not transmitted by this white flies. The gummosis disease of Phytophthora caused by Phytophthora palmivora in citrus is identified by the observing. Generally, first of all, I will tell you something. This actually the citrus gummosis is known to caused by 6 species of Phytophthora. Phytophthora citropthora, Phytophthora palmivora, Phytophthora hibernalis, Phytophthora syringae, Phytophthora cactorum. So there are more than 6 species are there. While I was doing my UG, our professors used to tell Phytophthora citropthora is a very important name, and in South India, if you see, it is Phytophthora palmivora is known to cause the disease. But all the other species, the most recommended species and identified species in all the cases is Phytophthora citropthora and Phytophthora palmivora. So generally, I studied at Phytophthora citropthora, since it is mentioned Phytophthora palmivora is also right. So they're asking the identified observing, which means a specific symptom. Root rot, chlorosis, death of shoot tip, and oozing of gum. So the name itself indicates gummosis, which means a gum-like ooze or gum-like some kind of liquid kind of material is exuded from the infected stem region, just above the soil region, maybe 30 centimeters or maybe 40 centimeters, it depends. So just above the soil region, we can see just gummy kind of ooze. So that is a most important character symptom of this gummy citrus gummosis, which is caused by Phytophthora species, Phytophthora citropthora, Phytophthora other species are there, right? So which of the following is a typical blue stain fungus? Polyporus, Ganoderma, Ceratocystis, and Penicillium. Probably so Ceratocystis is a blue mold. There is a fungus called Penicillium italicum, I think. That is a blue mold fungus, but the blue stain they're asking probably Ceratocystis, I think so. So which of the following is not a double-stranded DNA virus? So which of the following virus is not a double-stranded DNA virus? Cauliflower mosaic virus is a double-stranded. Bean golden mosaic, I don't think so. Rice tumor virus double-stranded. Soybean chlorotic probably double-stranded. I think a bean golden mosaic virus is not a double-stranded virus, I think so. Simcast is a computer simulation model is used to forecast lead beta potato, early blight potato, wheat stripe southern corn leaf blight. Probably Simcast is a model that is actually developed for this lead blight of potato, which is caused by Phytophthora infestations, right? Which of the following is the causal organism for hot rot of coconut? I repeat, hot rot of coconut in palm, algae, protozoa, virus, and fungus. This disease is caused by protozoal disease. Actually forgot the name. Hot rot of coconut. Anyway, I will just comment it below, or if you guys also just comment below if you know. In many fungi, vegetative hyphae of some same colony are of two colonies of the same spore coming in exact with each other, often fuse and the fusion called. So they just very simple. This fusion of hyphae is known as. They simply mentioned that two colonies are the same spore where they are in contact with each other and they are fusing each other, right? Very simple. Hyphal fusion. It generally is known as anastomosis or hyphal anastomosis. The answer is C. So sometimes they may, they will just extend the question, or sometimes they may give in a tricky way. Very simple here. They are simply asking fusion of hyphae is known as. Fusion of hyphae is known as anastomosis or hyphal anastomosis. So uh, which one of the following belongs to the kingdom Chromista? Plasmodiopora, Polymyxa, Spongospora, Phytophthora, right? Among the given options, this Chromista, right? So this Phytophthora. So Chromista means this Oomycota only. This Plasmodiopora comes under this Protozoa, right? So the answer is Phytophthora. Phytophthora, Pythium, all downy mildews comes under this Chromista, that is Oomycota group of fungi, right? A number of aphids in the field caught from the field are tested for the presence of virus serologically with. Very simple. You are collecting some kind of insect vectors, that is aphids or whatever the insect vectors, and you are bringing to the laboratory. And what are the which are the following tests you recommend for the identification of viruses? Among the given option, the best one is ELISA. ELISA is the best recommended for the identification of plant viruses. Because of this sensitivity. It is the most sensitive technique used for the identification of plant viruses. So the recommended tests, whatever the insects or whatever the stuff you collected from the field, we can go with ELISA. The stimulant in the root exudates of tobacco, which induces the germination of Vorabanki seeds, benzobarin derivatives, methyl bromide, allyl alcohol, and dazomate. So which of the following is in the root exudates of tobacco that actually they are telling that helps to activate the tobacco. So generally this tobacco roots produces benzobarin derivatives. These benzobarin derivatives are highly favorable. It acts as like some kind of germination kind of stuff for this Vorabanki. So generally is plenty of seeds which are actually from the previous seasons were there in the soil. So where this tobacco roots produces benzobarin derivatives, with the benzobarin derivatives act as the germination factor for this Vorabanki seeds. So if the tobacco cannot produce a benzobarin derivatives, probably we can suppress this Vorabanki, but there is no such kind of varieties have been developed so far without benzobarin derivatives. Generally this tobacco produces benzobarin derivatives only. So that benzobarin derivatives only helps to germinate these seeds. The next one is a molecule produced by the pathogen which induce a response in the host: toxin, elicitor, patotoxin, and PR proteins. Generally these elicitors are the molecules which are produced by the pathogens that actually triggers the defense response. We can say PTA, and again it is a very big thing. PAMP triggered immunity, effector triggered immunity. There's a lots of stuff was there to discuss. When we are also planning to start a molecular plant pathology. There we will discuss in detail about this host pathogen interaction. What is PTA? What is PAMP? What is DAMP? What is ETA? Why this many layers of defense will be there? What is SAR? What is ISR? And what is PR protein? All those things we are planning to discuss there. But generally, the molecule which are produced by the pathogen that actually responses or induces the defense responses is elicitor only. It is a kind of molecule which is produced by the pathogens. Okay. Porins are present in the bacterial cell wall component. Generally, porins are the present in bacterial cell wall, that are actually secreted by where these porins are actually present in the bacterial cell wall component. They are asking LPS layer, EPS layer, cytoplasmic membrane, and mesosomes. Generally, these porins are present in a LPS layer. Next one is multiplication of virus happens within epidermal cells, plasmodesmata, phloem vessels, and xylem vessels. This multiplication of virus takes place in generally epidermal cells. A computerized software system called field runners is used to measuring the chlorophyll content of the infected plants, recording the flight of insect vectors, sampling of plant diseases, and all the above. So generally this a software field runners is used to just collect the samples from the plant diseases. It has no way related to this observing the flight of the vectors or chlorophyll content, all those things. Just this software field runners is used to collect the samples. That's it. Isomerase enzyme patterns in fungal cell is used for differentiating races, differentiating strains, measuring spore densities, none of the above. Generally, this isomerase enzyme patterns is used to differentiate the strains or biotypes. The two flask technique used for the extraction of nematode is based on the principle of centrifugal force, whereas this two flask technique, the number of races of Hemilia vastastrix was identified so far. So it is known to that so far 32 races of Hemilia vastastrix was identified so far in the world. When it comes to Phytophthora, probably 82 races, right? It's been years I'm studying the same thing. Maybe probably new races uh, might be also maybe reported, right? A biocontrol agent used to control the root knot nematode, Pasturia penetrans, Pseudomonas fluorescens, Bacillus subtilis, Dactylis species. The best biocontrol agent recommended for the controlling of nematodes is just this Pasturia penetrans is the best biocontrol agent recommended for all kinds of nematode, especially this root knot nematode. Resistant variety of neck blast of ragi. So blast is generally caused by Pyracloria virus and Magnoportha grigia, right? So this blast is in ragi also caused by same Magnoportha species. Where there is a resistant variety called GPU28, this is recommended for this neck blast of ragi. Agrobacterium tumefaciens enters into the plant cells through stomata, wounds, leaf hairs, and stigma. So this agrobacterium tumefaciens, which causes crown gall diseases or galls, which generally enters into the plant cells through wounds made by the insects or maybe mechanical operations, whatever it is, generally enters through wounds. The next one is a primary source of inoculum of angular leaf spot of cotton. Seed-borne bacterium, bacterium present in the soil, bacterium transmitted through insect, none of the above. This seed-borne bacterium act as a primary source for angular leaf spot of cotton, which is caused by Xanthomonas campestris, Pathovar malvacearum, I think so, right? Angular leaf spot of cotton is also known as bacterial wilt of cotton, also known as uh, angular leaf spot, bacterial wilt. One more name was there, actually forgot. Anyway, the main source of the next question, the main source of inoculum of Xanthomonas citri, Pathovar citri, which causes generally citrus canker. Citrus canker is caused by Xanthomonas citri, Pathovar citri, right? So the main source of inoculum, they're asking, bacteria present in the soil, bacteria carried through seeds, infected twigs and old lesions on the standing tree, all the above. Generally, this infected twigs like propagating materials. Of course, seeds also it can transmit, but generally it is reported that the infected twigs and this old lesions are the main source of inoculum for this citrus canker. The next one is fungi belonging to the order Melanchoniales produces conidia with pycnidium, acervulae, sporacium and cinema. So this Melanchoniales generally produces conidia in a structure called acervulae. It is one of the asexual fruiting body like a Coltrotrichum, where it produces spores from this acervulae only. Virus with the split genome. So which of the following where we can observe the which of the following virus where we can observe the split genome? And I think in previous question papers also we might have discussed this. This wound tumor virus is known to having this split genome. I still not very clear about this. What is the split genome and all that stuff? Anyway, I will produce in the further editions. So the answer is wound tumor virus. The nematode organs which have important functions. I repeat, the nematode organs which have important functions: amphids, ampulla, bursa, and none of the above. This probably amphids. I am not well versed about this nematology, because while I was studying UG, PG, we don't have a separate section for nematology. I think we might have studied in UG, but we never concentrated in detail about this nematology. Now this nematology shifted to plant pathology. Again, there is no way. Again, we need to study. Anyway, we're also planning to do videos on nematology. Soon we will generate a good content on nematology as well. So the answer is amphids. The incubation period for stem rust of wheat in South India: 30 days, 27 days, 12 to 15 days, and 20 days. Generally, it is known to be 12 to 15 days is the general incubation period for the wheat stem rust in South India. Ammomum subabulatum is one of the alternate hosts for the following disease: leaf curl of tomato, cutter disease of cardamom, coconut wilt disease, grapes anthracnose. Generally, this cutter disease of cardamom is known to be this Ammomum subabulatum is act as an alternate host. Okay. Tip over disease of banana is caused by tip over disease of banana is generally caused by Irvinia keratovora. Out of 25 viral diseases recorded in India on potato, which of the following is the most destructive disease of potato? Leaf roll virus, rugose mosaic virus, potato virus X, potato virus Y. Before it is potato virus X and potato virus Y. Now in the recent days it is evolved as rugose mosaic virus is causing huge losses on potato. So the answer is rugose mosaic virus. Curzate, a systemic fungicide, is specific for the following disease. So curzate is a systemic fungicide that is recommended for the which disease they are asking? Downy mildew, powdery mildew, smuts and wilts. Generally it is recommended for downy mildew diseases. CaRNA5 is a just generally satellite RNA, satellite virus, bacteria. Generally it is a satellite RNA. CaRNA is generally recommended for satellite RNA. Yellow mosaic of legumes was the first reported in India from Shimla, Solan, Delhi and Kanpur. It was first reported from Delhi. Most commonly used transporter used in molecular plant pathologies. So TN5 is the most recommended transporter that is used in molecular plant pathology. Wild host of same families infected by the pathogen are called. I repeat, wild host of same family. So alternate host is different family. Collateral host is same family. Collateral host is also known as alternative host. Reservoir of the pathogen, subsidiary host pathogen. Generally, alternate host is actually belonging to other families. For example, you can take wheat stem rust. Wheat is one family and barberry is another family. When it comes to collateral hosts, where the primary host and secondary hosts are belonging to the same family. For example, you can take rice blast. Rice is Poaceae and the grasses, let's say Exandra, which harbors the pathogen, is also belonging to the Poaceae family. So the answer is a collateral host. So same family, say they asked, right? Aprosoria contains following chemical for creating the turgor pressure that allows the penetration peg to puncture the cell. Generally, this aprosorium contains high amount of glycerol that can create high turgor pressure or pressure to make the punctures into the epidermal cells or cells. An example of trimethylamine producing a bunt fungus. We know very well the classic example is this is kernel bunt of wheat, which is caused by Tilletia indica or Neovicia indica. This is generally Neovicia. So the answer is both Neovicia and Tilletia, but generally Neovicia we used to study, right? And this wheat bunt is also called as the Neovicia, caused by Neovicia, generally caused by Neovicia indica and Tilletia indica. Mitra generally named it as a Tilletia indica. Later Munkur changed it to Neovicia indica, and later became controversy. And now we are using the both name Neovicia indica and Tilletia indica. The accumulation of ornithine. I repeat, accumulation of ornithine is due to type-toxin, fasciolotoxin, cercosporin and tentoxin. Generally, this ornithine accumulation is due to fasciolotoxin. Bud nest fungus was first mentioned by, I repeat, bud nest fungus was first mentioned by Carolus van Linnaeus, Clusius, C.H. Parsoon and Tulsen brothers. So regarding this bud nest fungus, it was first mentioned by Carolus Clusius. DAS used as a virus inhibitor under the laboratory conditions. Ethylene oxide, lactic acid, propylene oxide and formaldehyde. Generally this formaldehyde is used as a virus inhibitor in the laboratory conditions. The term inoculum potential was coined by E.A. Gauman. Woodbine leaf crinkle virus is a internal seed borne, external seed borne, both internal and external, none of the above. It is known to be that this woodbine leaf crinkle virus is a internally seed borne virus. Hypovirulence carries the genome. Generally, we know very well this hypovirulence carry a genome called a double stranded RNA. Indicator plant for striga. Which of the following is an indicator plant for striga? Cotton, peanut, tomato, both A and B. Generally, this cotton and peanut both is known to be the indicator plant for striga. Thermal death point of tomato spotted wilt virus. 45 degrees centigrade is the thermal inactivation point for tomato spotted wilt virus. Woodbine leaf crinkle virus is transmitted by aphids, whitefly, seedborne, all the above. So it is known to be that all the factors are involved in the transmission of this woodbine leaf crinkle virus. Which of the following is a monocot powdery mildew diseases? Levolula, Ansonella, Phylactinia, Blumeria. Generally causes Blumeria, which causes powdery mildew in cereals. Cereals are mostly monocots, right? So where this Blumeria infects mostly cereals. Botrytis cinerea, which is mold fungus. enters into the plant cells through lenticels, hydatodes, stomata and nectaris. Generally hydatodes, it enters into the plant cells through hydatodes. Which disease uh, is considered as jaundice of wheat? Wheat stem rust, wheat leaf rust, wheat stripe rust, kernel bunt of wheat. Generally this stripe rust is considered as jaundice of wheat. Wheat blast was first reported in, so among the given options, wheat blast was first reported in Brazil in 1985 probably. There is a area, Brazil somewhere, Panama, no, not, it's not Panama. Panama is physiognomy of banana, but probably somewhere in Brazil where this wheat blast was first reported. Temperature for lamp, loop mediated isothermal amplification, which was first reported by Notami with Aiken Chemicals Private Limited, where the main difference between the conventional PCRs and LAMP is where PCR ran at three temperatures: denaturation, annealing, and extension. So that ran at three different temperatures, but the LAMP can avoid all the temperatures. I mean, three different kinds of steps at maintains at one temperature, that is 65 degrees centigrade, where the LAMP runs at. Right angle mycelium is produced by generally produced by rhizoctonia. Okay, answer is rhizoctonia. Here it is, I think so. Endoconidium is producing fungus. Which of the following is an endoconidium producing fungus? Thylapsis bassiocola is known to be an endoconidium producing fungus. Apple scab was first reported by E.M. Fries. Birdness fungus was first mentioned in which one? Previously we reported that Clusius wrote a book called Rarorium Plantarum Historica in probably 1601. So this birdness fungus, where is Clusius mentioned in his book called Rarorium Plantarum Historica. Mineral oils is used against as an fungicide agent for mineral oils are Cercospora Muse, Nicotiana, and Botrytis obtusa. I don't know, probably Cercospora species, something as a fungicide agent. Anyway, please hold this 68 question, or else if you guys know, please let me know the answer about this 68. The next one is 69. I'm going. Wheat flag smut is inflorescence smut, culmiculus smut, seedling smut, ovariculus smut. So it is known to be wheat flag smut is a seedling smut. Kumbu smut is an inflorescence smut. Wheat loose smut is a ovariculus smut because it infects ovary. So based on the parts they are giving the names, nothing else. Slime spore is produced by which of the following produces a slime spore? Probably fusarium produces some kind of slime kind of spores, right? Whereas root rot, sclerotium, all those things will produce, but fusarium produces slime kind of spores, right? Entomoptera muscae is a ink fungus, hair fungus, pinmold fungus, fly fungus. So Entomoptera muscae is actually a fly fungus, right? It can kill the insect by flying, right? Cardamom mosaic virus was first reported by Molinson. Cardamom mosaic virus was first reported. Citrus cissula virus was first reported by Brown. Collateral host for cutted disease of cardamom. Collateral host, I think we have already discussed, right? Black cardamom. Collateral host, okay, asking collateral host. Black cardamom, hill cardamom, then all the above. So all the above are generally the collateral host for this cutted disease of cardamom. Buddiness fungi belonging to the order Nidulaceae. Cormium producing fungus. Which of the following is a cormium producing fungus? Penicillium claviforme. Nepo and Netu viruses having bipartite genome, single stranded RNA, bipartite single stranded tripartite. Okay, the answer is a bipartite genome with a single stranded RNA is the best example for Nepo and Netu viruses. First reported virusoid. Which of the following is a first reported virusoid? Subterranean cloud, Lucerne, Solanum, Nidifolium, Valvet. Yes, the first reported virusoid is a Valvet tobacco mottle virus. It's not viroid, virusoid. They're asking Valvet tobacco mottle virusoid. Okay. In recent ICTV classification, International Committee on Taxonomy of Viruses, TMV, tobacco mosaic virus, actually belonging to the order. So before it was Tobamaviridae, now it changed to Virgaviridae in the recent classification. Most of the plant pathogenic bacteria are rod gram negative, rod gram positive, filamentous gram negative. Most of the plant pathogenic bacteria are rod shaped gram negative bacteria. Okay, Which one is a monocyclic? Wheat, loose mud, rust, powder mildew. Okay, wheat loose mud is generally considered as a monocyclic. Following is not a general elicitor. Not a general elicitor. Glucan, hairpins, flagyellin, AVR. So generally this flagyellin is not a general elicitor. PR5 is related to. So PR5 is thematin-like protein. There are 17 kinds of PR proteins that have been evaluated. PR1, beta 1, 3 glucanases. There are some kind of thing will be there. Generally, this PR5 is a thematin-like protein. ITS stands for generally internal transcribed spacer, which is the most common, uh, most common primer used in sequencing of or DNA molecular identification of this fungi. Okay, shape of a bacterial growth curve. Generally, this bacterial growth curve are sigmoid in shape. Zygospores are formed by. So zygospores are formed by gametogamy, gametangiomy, gametansomatization. Okay, we can observe in rhizophus, right? You can just take an example of all zygomycota fungi where they produce zygospore with the process of this gametangy, right? Cowpea mottled in sexual reproduction. If we observe this rhizopus also where this gametangy process will happen. Cowpea mottle mosaic virus is transmitted by white flies. Aplonospores are produced by rhizophus. Aplonospores means non-motile spores, where Albogopithium phytophthora all produces motile spores, that is called juice spores, right? Coconut lethal yellowing is caused by coconut lethal yellowing is a phytoplasmal disease. We know very well, right? Dicer is actually related to RNAi, 16S or RNA, mRNA, ATS. So RNAi is actually where the dicer is related to, where this dicer kind of things can be observed in RNA interference, right? DAPI or DAPI staining is very specific for identification of RLOs, MLOs, PPLOs, bacteria, generally MLOs, that is Phytoplasma and Spiroplasma, right? So diameter of petri plate, generally we know very well the most commonly recommended or using petri plate diameter is 90 mm or 9 centimeter in diameter. Which one is not obligate in nature? Which of the following is a rust, smut, powder mildew, root rot? Rust, powder mildew, smut all are obligate only, but when it comes to this root rot, it's not obligate, right? It's saprophytic, generally saprophytes, right? So which one of the diseases is caused by the fastidious bacteria? They're asking fastidious vascular bacteria, black rot of cabbage, citrus canker, coconut kadam, citrus greening. Generally, this is citrus greening is known to be the fastidious vascular bacteria. The two phloem limited gram negative, I think so. Phloem limited gram negative bacteria, citrus greening, Candida salivary bacteria, asiaticus. Okay. Inverted cup shaped fruiting body is produced by which of the following? In Pycnidium, Peridium, okay. They're asking inverted cup shaped, right? Where if you see in wheat stem rust fungi, and the upper side Pycnidium is produced, and lower side Asium is produced generally. So inverted cup shaped there. So the answer is Asium. First plant pathogenic RNA virus to be completely genome sequenced. So for the DNA virus, 1980. RNA virus means tobacco mosaic virus, which was sequenced in 1980 to Golet et al. Right? Which are the following diseases not observed in India? I was still doubted about this. It is mentioned that fire blight of apple is still not reported in India, but I don't know whether it's true or not. Recombination due to mitotic crossing over called as parasexuality, heterokaryosis, karyogamy, and plasmogamy. So recombination due to mitotic crossing over it is generally called as parasexuality, which was first reported by Ponte-Carvo and Roper in Aspergillus nudulens, right? So these are all the answers. For further more informations and various kinds of other exam preparation, students can refer this book, A Vision into Plant Pathology, a complete student version. For further doubts, guidance and clarification, student can reach us at www.geekyresearcher.com. Stay geeky and stay tuned. We are Team Geeky Researchers.